In this video, we will demonstrate how GenePattern Notebook seamlessly integrates Python with GenePattern in a notebook by allowing you to use Python variables as inputs and outputs of GenePattern analysis cells. We will show how to use Python variables as input parameters to a GenePattern analysis cell, and how to send GenePattern analysis results directly to a Python variable or data frame. This tutorial assumes that the user is familiar with the basic concepts of both GenePattern and Python. Please note, however, that one need not know Python to use GenePattern or the GenePattern notebooks. Introductory tutorials for GenePattern notebooks can be found in the Notebook repository, notebook.genepattern.org, in the Public Notebooks tab. To begin, let's examine how you might use a Python variable as an input for a GenePattern module. In this example, we have a floor value, which we want to pass into GenePattern as the floor value in our pre-processed dataset module. In order to pass this value to GenePattern, we create a variable. To now use this variable in a GenePattern module, we must insert the GenePattern module into our notebook. To insert a GenePattern module, we select a new cell, then change its type to GenePattern. This opens a menu which allows us to select and run GenePattern analyses on the GenePattern server. We're going to use pre-processed dataset, so we search for that module, then click on it to insert the module into the notebook. We can then provide the floor value as the variable we just defined, using double braces to indicate to GenePattern that this is to be read as a variable. To see how the GenePattern widget is being rendered in Python in the notebook, you can toggle the code view. To do so, click on the gear icon and then select Toggle Code View. To run, simply click Run as usual. To use the output of a GenePattern module as a Python variable, click on the output file name or the info icon next to it and select Send to Code. From here, you can now manipulate the data in Python as you would any other data of that type. You can call help on the job ID to get more information as to what functions are available for working with GenePattern output files. Another Python integration we've built in is the ability to send GCT and ODF files to a pandas data frame, and thus display the data matrix in line, plus all of the functions for a data frame. Note that the pandas library is pre-loaded in our hosted repositories. As a quick reminder, a GCT file is a tab-delimited file that describes expression data. The first line defines the format version of the GCT file, which should be 1.2 for all current GenePattern modules. The second line defines the number of rows of data and number of samples. The third line contains the names of the samples. The first column defines the probe or gene IDs. The second is an optional description of those identifiers, and the rest of the columns contain the expression values. ODF is another data matrix format. To display the GCT file in a data frame, select Send to Data Frame. This will generate the data frame code, which, after it is executed, will display the GCT file in a pandas data frame, and you will see the same matrix of data with GCT-specific formatting stripped out. This concludes our overview of the GenePattern Notebook Python integration. More comprehensive code examples are available in our example notebooks, which can be found in with other public notebooks. We specifically suggest the GenePattern files in Python tutorial. For more information about GenePattern notebooks, please visit genepattern-notebook.org.